Thank you. So welcome everybody to my presentation. As you see from the names, I am coordinating a larger group here at the University of Pavia, but also with other collaborators. And I especially want to mention two PhD students who have worked on the research on recreational boating in the past 6 years, that are Dr. Jasmine Ferrario, currently a postdoc in our university, and Dr. Eileen Ullman, who currently leads a private consultant agency in Turkey, but they have worked an incredible amount of hours on this project. So I'm going to talk about my biogeographic region, the Mediterranean Sea. It is a small sea, but hosts an incredible diversity of cultures and languages and history also. So it is considered a hotspot for tourists from all over the world who are curious to see this cradle of civilization we have. But unfortunately, the Mediterranean Sea, it is also a world champion in marine bioinvasions. As you can see from this graph, we have 800 multicellular marine NIS, non-indigenous species, mostly are in the Eastern Mediterranean Sea, where the main vector of introduction is the Suez Canal, this artificial waterway that connects Mediterranean Sea and the Indo-Pacific region. But we also have all the major vector of introductions that are very well known elsewhere, that is aquaculture and shipping. And in particular, hull fouling seems to be a big problem here. From the legislational point of view, the problem of marine bioinvasions has been acknowledged quite a long time ago already, 15 years ago, by the Barcelona Convention, which is a regional sea convention by the EU Environment. In 2005, in fact, this Barcelona Convention produced an action plan concerning species introduction and invasive species in the Mediterranean Sea. And hull fouling was already acknowledged as a vector, and this action plan encouraged the development of a regional plan on hull fouling. This action plan was then updated 12 years after, in 2017, and in the updated action plan, there are national plans encouraged to prevent vectors. So you can already imagine that not much has been actually done in the meanwhile. In addition to this, we have also legislation instruments from the European Union, because 8 Mediterranean countries are part of the European Union, so are subject to its laws. And in particular, I want to mention the Marine Strategy Framework Directive that promotes monitoring, especially of high-risk areas. And the more recent regulation on invasive alien species from 2014, which promotes prevention and control on a specific subset of invasive species. At present, no biofouling species are included in this list. So, with all this background, the reality is quite different. So, the monitoring in high-risk areas and even on biofouling species is at very initial phase, and there is no agreed protocol even within countries. Different regions do different things. So there is not a... We are still very far from achieving a baseline monitoring of this region. Prevention and control of biofouling do not exist at all at present, or not as I am aware of. The Mediterranean Sea however, has to be considered with caution because it has a large traffic of recreational boats. 20% of the global boat fleet of the entire world is in the Mediterranean Sea. It means that one boat out of 5 in the world is in our sea, and it's because the Mediterranean is rich of landscapes, vegetation, cultures, archaeological sites, and we also have a very long tradition of boating. In fact, my country, Italy, is the world leader of the production of super yachts, and there are many, many boat yards, haul-out facilities, so this is a boating paradise. So there is quite a large traffic. However, it has never been studied in detail as a potential vector of non-indigenous species transfer. So what we did was to try understanding the role of recreational boating as a vector and the role of marinas as possible hotspots of introduction. We tried to investigate what are the factors, abiotic factors, that make some marinas at higher risk of introduction than others. We also wanted to investigate the behaviors of boaters, how they travel, how often, how they maintain their boats through interviews. And we also studied the hulls, so what's under the boats. So all these studies took us 6 years, nearly, and we used a variety of approaches and methods here. I am only synthesizing a larger amount of work, obviously. So what we did was surveying ports and marinas. We did the scrapes with this net that you can see in the photo on the right. We scraped the docks, pontoons, any floating objects, looking for the alien species that were hiding underwater. And also explored the hulls of the boats, either underwater by snorkeling or diving where possible, or at the boat yards, so when the boats were taken out of the water for cleaning or painting, and we explored their hulls. We also submitted questionnaires to the boat owners regarding their travel history, maintenance, and also we wanted to explore their awareness on the problem of alien species. And then after collecting all these samples, we did a huge work of species identification and assessment of presence of non-indigenous species. In the marinas we visited, we also recorded some biotic factors. Some are related to environmental features like salinity, temperature, climate characteristics, chlorophyll, and so on. Some factors that are related to the structure of the marina, like the number of birds, the size of the marina, the presence of shipyard, the presence of floating pontoons, and so on. And also recorded proximity to other potential vectors of introduction, like aquaculture, commercial harbors, and the proximity to the Suez Canal, that, as I told you, is an important pathway of introduction in the Mediterranean. In this slide, you can see our study area. We tried to sample the northern rim of the Mediterranean Sea, visited 7 countries, and all this work has been done by the 2 PhD students I mentioned you above. But this study area was divided in 3 parts, basically. So in a first experiment, we wanted to compare the richness in non-indigenous species of recreational marinas and large commercial harbors in the neighborhood, because we hypothesized that the pool of non-indigenous species in a marina is a subset of the non-indigenous species that is in a larger harbor. So we hypothesized that the larger harbor is the first entry point for non-indigenous species, and the marina was the site of secondary dispersal. So we sampled 5 harbors and nearby marinas in the northwestern Italy and Sardinia. Then, for the study of correlation between abiotic factors and NIS richness in marina, we consider the whole data set, so all the 50 marinas of our data set. And for the study of boat hulls, we only consider 25 marinas that were those where sampling the boat hull was feasible, because in some marinas we did not get permission to go sample the hulls of the boats. In total, we sampled and interviewed nearly 600 boats, which corresponds to approximately 25 per marina. So these were the 3 stages of our work. Let's move to the results quickly. First stage, comparing harbors and marinas. And first surprising result for us. We expected marinas to be, as I told you, to have a subset of NIS compared to ports, but this was not the case in all the sample localities. So there were localities where the number of NIS was comparable or even higher in the marinas, and even the ratio between NIS and native species was comparable or higher in marinas. And surprisingly, some NIS were unique to marinas. They did not appear in harbors, but were occurring in marinas. So this was published in 2017 on a journal Biofouling, and here we showed that actually marinas can be considered hotspots and even primary sites of introduction of non-indigenous species. Following this, we carried out the larger assessment. And in this map, you can see how we were overwhelmed by non-indigenous species when we analyzed our samples from 50 marinas. We found 74 NIS, from 2 to 27. The marina with the higher species number, 27, was here in the island of Crete. As you can see, non-indigenous species belong to a variety of taxonomic groups, and there was no marina without at least 2 species, 2 non-indigenous species. And these species were even new for the Mediterranean. 3 of them were totally new, never seen before in the Mediterranean. And we also managed to provide 51 new country records, meaning that some species were already known from the Mediterranean Sea, but not in a specific country. And this was published in 2017 on the journal PRJ. This was a very, very long and large paper where we described all these massive number of records that we found. Okay. Some of the species were only locally distributed, but some other were very common across the Mediterranean Sea. As you can see, some occurred in over 50% of the sampled marinas. and some of them were already very well known from the area, but some species like Sellepuraria brunea or Parantura japonica have been reported only in the past decade from the Mediterranean Sea. So you see they have apparently spread very fast in just one decade over the marinas of the Mediterranean Sea, meaning that this continuous transfer of species is happening now. As regards the factors, the abiotic factors that we found significantly correlated to NIS richness, we found temperature, number of birds, distance to Suez Canal, and then proximity to other vectors like aquaculture, commercial harbors, presence of floating pontoons, and biogeographic sectors. So these are the factors that seem to be best related with the risk of having many NIS in a marina. We are now trying to investigate further on this aspect and develop a more quantitative model. These results were published last year on the Journal of Environmental Management. Finally, third step, the analysis of recreational boats, hulls. This is probably the biggest replies we had. In fact, we noticed that out of nearly 600 boats sampled, more than 2/3 bring at least one non-indigenous species. It's 71% of this sample of boats that we collected from different ports, from larger marinas, smaller marinas, eastern, western Mediterranean. So it means that the boats that are moving in the Mediterranean are really highly responsible of this non-indigenous species transfer. And we found up to 11 non-indigenous species on a single boat. And even more worryingly, we found that even boats that were cleaned very recently were hosting some non-indigenous species. In this slide, you have a table, but this is just an extract. I have a much longer table of cases but these are all rows are single boats, and then this is with indication of the marina where we sampled them. And as you can see, even in boats sampled 1 week ago, there were 3, 4, 5 non-indigenous species. And this is really worrying, because then even a frequent cleaning is not enough to prevent the problem. When we looked at all the data set and tried to relate the presence of NIS to factors related to boat maintenance or boat features, we found these factors to be significantly related. So factors like the time since last clean, since last paint, and the number of days spent traveling were related to NIS richness, along with the boat length and average cruising speed. And also the percentage of visible fouling in the hull and in the niche areas of the hull were significantly related with NIS richness. So we have some factors that can be considered for management. We also interviewed the boaters, as I told you, about their traveling habits, and the map that you are seeing now reflects the interviews that we did. This is the real data that we collected in the 25 marinas, asking boaters where they have been in the past year. And as you can see, the movement is noteworthy, and boats get in and out the Mediterranean Sea from any possible waterway, from the Straits of Gibraltar, which is a natural opening, to the Dardanelles, that is opening towards the Black Sea, the Suez Canal, that's the artificial opening towards the Red Sea, and even in the inland waterways towards Atlantic coasts of Europe. So there is a huge movement. They travel pretty much because they spend a relevant number of days outside their own marinas, and on average visit 7.5 other marinas per year. So we have boats traveling a lot and bringing a lot of non-indigenous species. So this cannot be ignored anymore, I think. The results on the study of the boat fouling was published just a few months ago on the Journal of Applied Ecology. Something about the awareness, because this is also a problem. The awareness we asked the interviews, the boat owners, if they know alien species, and they asked. And we asked them to name us one alien species. So the results are not very comforting, because the general knowledge of the problem of alien species is often insufficient. And when they are asked to name an alien species, some of them correctly mention one. For the most popular species resulted the so-called killer algae, Caulerpa taxifolia, which is an aquarium introduction in the Mediterranean Sea, and the rabbit fish Siganus luridus, which is a Red Sea immigrant. None of them mentioned any biofouling species as alien. And then some of them mention weird species like the bonefish or the monk seal, which is an endangered endemic Mediterranean species. So their concept of alien non-indigenous is very, very approximate. And this makes a problem, because if they do not perceive the issue, they will be difficult to involve in prevention strategies. So another challenge to address this problem is monitoring, because as I told you in the beginning of this presentation, in the Mediterranean Sea, we have no agreed upon or standard protocol for collecting, identifying, and quantifying non-indigenous species. And we also have a taxonomy problem. So in the Regional Environmental Agency, there is often no personnel able to identify non-indigenous species from the fouling community. And this is the largest challenge, probably. Prevention and control, we haven't probably even started to think about this. Without awareness and without monitoring, it's difficult to draft reasonable prevention or control plans. So with this optimistic note, I end my presentation here.